Hello! In this video, I am going to go through the week 4 Thursday script file for fall 2024. So there was a few caveats here. I was just trying to ensure that you were paying attention to this being a linear process. Generally, also when you're data cleaning, since it is often iterative, you really want to make sure you're carefully watching how the object changes as you're progressing, and that it should be a progression. You shouldn't be going up and down depending on how things worked out. You should just be going step by step. Eventually, you get this and you get these scripts so that you can just run them. You run them, they work a particular way. You can maybe even eventually modify them so that you give them certain inputs and they just do the same thing every time. In fact, that's kind of what we're working on next week when we start writing functions, which are essentially just that. They are objects that have a set of directions inside of them. And so you give them some inputs and they apply that process to your inputs. And so that is doing a certain piece of code. All right, so that much philosophy aside, that culminates to, you should probably remove the objects. Now, I just want to point out, when I run this, all of my data in the environment is going to disappear. Not this. These are, well, you can't see it. These are files stored inside of my home directory, but those are stored on the computer in a permanent or semi-permanent storage. Objects up here, so things like I have created, here I'll make iris. I was going to make iris 2, but before I couldn't type. I just remembered there is an iris 2, so I shouldn't have done that, but oh well. But let's say I did that. Hey, I shouldn't have done that. Let me remove these things. And let's say I made a iris 3 and I don't know, irish. I have all these objects I've just made. I could click that little cleaning button, or I could run this handy piece of code which first lists out everything in my, right there, the list ls function, which lists out all of my objects that exist in my global environment, and then I can remove them, having to send that input to an argument titled list inside of remove. I know you've seen a lot of these names before, and even here it's a little bit redundant. This is listing, a verb. This is list, as in it's a list of values, even though it's actually a character vector of values. I think this is kind of an old carryover. This probably existed since the early days of R, and they hadn't quite settled on the language yet. But anyways, you run it, all of my stuff disappears. I'm ready to move on. That's why you shouldn't have done that until you were ready. I spent more time on that than I meant to. Next, I'm going to set my working directory, because that's always a good move. When I do that, it means that if I start typing in something like read.csv, and then I open up a quotation marks and I press tab, the nice thing about having set my working directory is my file right there, or nope, right there. So I can then press tab again, ending that, ending the quotation mark, ending the parentheses, can run this. It says that's not quite right. I start to then have to figure out all right, how do I debug this? Well, I can currently see that it's because this is a TSV, tab separated values file. But let's pretend I wasn't sure what was going on. I could remember that Nolan said that there is a tool to help us import data. So I'm going to go here. I'm not going to pick readr. That's a separate package. I've asked you not to use readr or other packages unless I tell you to do so, at least until after the midterm. So I'm going to pick base. I'm just going to open it up in base R. At that point, I'm going to select the file, open it. It's now visualized. I can see, oh, the problem is that these are tabs, not commas. CSV won't work. CSV is for comma separated values. These are tab separated values. So this needed to be tab. It actually automatically did that for me. It even found that I wanted a header. I could have told it no, but that would be a bad move, because now these numbers are being combined with some letters, and that means it's all going to be characters. That's a whole problem. I want those to be numbers. I want those to be headers, characters, heading. This one's not numbers, it turns out, but we'll learn that in a minute. Usually the last thing I'm going to do up here is I may change this, although it really doesn't matter. I could do a bunch of gibberish if I wanted to. I say that because I want to point out that all it's doing is helping you write the code. I don't have to agree with how it wrote the code. It's helping you write the code. It's helping you write things like the path, and if there are any arguments that need to be set. In this case, I didn't have any that needed to be set. It had a heading. The default behavior is have a heading, and so no arguments need to be set. I could now go in and start changing things. I could say, don't load it with a heading, but that would do a poor job, and I wouldn't get, my nice column names if I do that. So instead, I'm just going to leave it as it was, or leave this at least at true. And that way, when I come over here and I test it out, look at those beautiful column names. That's a nicely structured object. I can even see that that first column is in fact numeric. Integer column didn't quite make it though, so we're going to have to do some cleaning, some character values, some video game names, and the character value, character. These all had letters in them. The only one that was solely composed of numbers, and so that actually got imported as numeric, is the random column, which contained a bunch of random values. I actually generated those. All right. Next thing, it said make the column names lowercase. So here would be my answer for that. This is a demonstration of how we can assign to certain functions. Oftentimes, those functions are actually things like extraction operators, but it does also include certain functions that are built to do this, to allow you to assign to them. So here, what I'm demonstrating is that if I pull out the column names of studf, I can assign to those names to overwrite them. Not only that, is by pulling them out, I can also do functions on them first. So here, that's exactly what I do. I first call this data, so these letters, which have a bunch of capitals in them. I then change those to lowercase values. And then finally, I assign those new values, overwriting the old values. So I've assigned it new values. Now, when I print it off, my column names are all lowercase, which is what I wanted. This next line, I don't know if this was a good idea. I liked it. I thought it made sense. Trying to get you to think ahead a little bit and just try to picture how some of these tools get used together. Here, though, a function is being made. So this is the first time you've seen this, I believe, in this class. I am actually making a function called symbol check. It's going to contain a single argument. That single argument is then going to be used here to produce a y. Y is then going to be used here to produce a z. And then I'm going to return, see, get it? Return. I'm going to return some data structured as a data frame. That's a combination of some information describing y as well as the contents of that z object that was created. All right. Well, we can play with this. It turns out that the easiest way probably to break this down is to first make x, and we can rely on the fact that one of our other assignment functions, operators, is the equals sign. 1 equals, not 2 here. When I did that, when I ran that line of code, which I did with a hotkey, it was added to my environment pane over here. It's just the contents of that column. So here we have a character vector named x. It's got a lowercase x. It's got a bunch of gene descriptions in it. These are coming from the Arabidopsis annotation. So the first thing I'm doing to this x object inside the function is I am splitting it. But I'm splitting it in kind of a weird way. I'm splitting it across no characters, which just has the fun result of splitting it between every single character. Now that's not maybe the most common, although that may be the most common, I don't know. That's a usage of string split, but I think a more understandable usage is if I wanted to say separate it by space. So here I've separated every description value by space, although we can see from how this is printing out, it's actually resulted in this object being stored as a list class object. Note this double square brackets. So here's container 75 that contains a 1, 2, 3, 4, 5, 6, 7 length vector of character values. So that's just how this works. The problem is it's kind of hard to do stuff like unique on this. It may work. I don't actually know for sure. No, unique did probably. Yeah, there's no way that's the first time it said DNA in this thing. So I'm thinking it's only returning unique values within each container, not across the containers. I'm just looking for the word DNA again. Okay, I definitely just saw RNA and RNA. So cool. And this is demonstrating how we ask ourselves questions in the experiment. I wasn't really sure, but that's why I use this unlist, is because applying functions to the data they're not intended to work on, so like a list type object, it behaves unexpectedly. However, if I first take my list object, I unlist it, that's going to make one big vector of character values. I can then call unique on that, and we're going to go from 308 values to 176. Now, I'm going to repeat that with the original data, and we went from, well, let's see. We went from 993 plus 1,833 characters into close to, what, 60 or so. Maybe a little bit above, like 69 or so. Let's see. Length. 62. Ah, was close. 62 unique characters, which are all in here. Now they're kind of in a weird order because this is based on the order that they appeared in the data set. And so the final thing before I made Y was I sorted it. So that was kind of a more understandable order. All right, so that was used to make Y. I can now experiment with Y. Cool, Y is that data I just visualized. It's then doing grepl. And this one I think it just helps to see side by side. grepl is going to be searching for the pattern alnum. That's something we've talked about, but the documentation, if you search for it, is going to show you that this stands for alphanumeric values. So it's looking for any alphanumeric character. Since these are all length 1 characters, then that means that if that character at that position is alpha or numeric, alphabet or numeric or numeral, it's going to return true. So those were all true, but the symbols, so here and to the left, all of these returned false because they are not alnum. How do you know that? You look it up. You wouldn't have known that off the top of your head. Or I guess maybe you could have guessed, but you didn't need to. Anyway, finally, the last step is that it uses, I had to actually run that line of code, and then finally that last step is it assembles all of that into a data frame and returns it to the user. Now, at this point, I'm just going to demonstrate good behavior. These little values can become kind of dangerous. Generally, if you are making functions in your global environment, you don't want to name the objects on the outside anything same as on the inside of the function. And the reason why is that something goes wrong in here, it will actually look into its parent environment, so here your global environment, to try and find that value. And that can be really confusing. And so this is just me giving some good advice because I've done this for a long time. You should clear your environment of any objects from the inside of the function. That should just generally be avoided. There are ways to protect against it. It's just a thing I promise becomes up. So I cleared my environment. I also could have just gone up, could have removed it. And then I just ran everything because I've been organizing my code in a linear fashion. Really easy to repeat. I got down here, it ran that custom function, and boom, there is the output. So none of those x, y, and z's, those are all inside the function only. On the outside, I just see this printed out message, and I see the functions object. So that's what's going on. It splits the character, unlists the resulting object, finds the unique values, and then sorts it. I then do true or false. Is it alphabet or numeric in value using that alnum grepl? And then finally, I return a data frame of the results of that analysis. Not too bad. Once you know all these different functions, there's a lot of functions. All right, next one, make a new column in studf called defclean, containing the values from the student data frame Arabidopsis definition line. It is literally this easy. Like I said before, the idea is that you can extract data and assign it new values. But a cool thing about the $sign tool is that if you extract a container that doesn't exist yet, you can put whatever you want into it. Now, if you try to put a wrong size object into a data frame, it actually ends up converting whatever you tried to add to a list class object. Because a list class object, they can be all sorts of different sizes. Remember, list type data is a series of containers. Well, list class object leans into that. And so each of its containers can be a different size. There's no problem with it. A data frame can only have containers of the same length, because that's how it allows it to be organized into a rectangle. But here, because I'm basing this new column on an old column, they're definitely the same size, so no problem. New column gets created, where once there was null, there is now, I don't know, a bunch of genes. That sounds very dramatic though, right? All right. Next up, replace the above symbols. I've got to change that phrasing. That was confusing everybody. But the symbols I'm referring to is these symbols. Sorry for the confusion. Replace them with spaces. So we could, now for the record, we could have done just a ton of G subs. We could have done one, and we could have done another one. So that could have been one, this could have been another one. And as long as we kept replacing and extracting from the same thing, so we're just like referring back to defclean, overwriting it, using the new version, overwriting the old version, and just repeating that, we could have done this without any ors. We even could have used the fixed argument and not even needed to worry about escapes. Ironically, the one I just picked didn't matter. That it doesn't need an escape hyphen, but my point stands is that we could have done a lot of different ways. But in order to do all of them at once, you really didn't have an option other than to write a fairly complicated pattern. Now here, I kind of just went ham with my escapes. Each escape has been placed in front of each symbol, making sure that that symbol is interpreted as the symbol, not as its special meaning in regular expressions, if it has one. So for hyphen, which doesn't have a special meaning, this actually doesn't change anything. I could have left it like this, and nothing would have changed, because hyphen doesn't have a special meaning. This symbol, a open parenthesis, is not true. Open parenthesis does have a special meaning, and it absolutely needed to be escaped. Same for closed parenthesis. Not true for comma, although comma can have special meaning in regular expression. Hence, the kind of like, just escape it if it's a symbol makes this much easier. Forward slash, I can't remember if it has a special, I don't think it has a special meaning, but you think it would. Semicolon, I believe, is again another kind of contextual one, but I think it generally doesn't, but I don't know. And finally, plus absolutely has a special character meaning, and so it almost certainly it certainly needed a escape. Now let's talk about the escapes real quick. What's going on with them? The escape, if you don't remember, is a symbol that tells R to not consider the next letter its normal meaning. The next letter, in other words, has a special meaning. Now here's the problem. grep also uses escape for the same idea, although it's kind of in the opposite typically. So for grep, it also uses backslash to say that the next character has a different meaning than normal. So for instance, a period, which normally in grep stands for any character, it needs to match instead with a literal period. It would need a backslash. But the problem is that if I say backslash period, I get an error message from R. That's the problem. This is coming from R, not from grep, but from R, or not from gsub, but from R. I get a message from R saying that backslash period is not a valid escape, and that's because R also uses backslashes as its escape. There's like 2 levels of interpretation. So this is the weird thing. I have to backslash my backslash. This first one is to tell R that, hey, this next backslash, send that baby on over to gsub, because that's not a special character. It's just a regular old backslash. Then gsub now gets this backslash, and it sees, you mean an actual period. Sure, no problem. It completes without an issue. This is how the double backslashes work. In the end, I mostly just remember, oh yeah, with regular expressions, there are multiple levels of interpretation of those escape symbols, so I just need to put 2. It does get weird when you need to put like 3 or even 4 backslashes, because you have to like backslash your backslash to backslash your backslash, so that can backslash another backslash for a 3rd level interpretation. I think maybe 5 I don't know, you get lost sometimes. It can be tricky. I've literally just sat there and put backslashes on occasion. Anyways, you'd go through this process. You can either just backslash them all, or you could sit there and play with it until you figured out the minimum number of escapes. I chose to just backslash all my characters, or all my symbols. And once I do that, I've replaced all of those with a space if it found them. And if I print that off, it should make sense. So I come in here, and what used to contain a bunch of hyphens and other symbols have been removed. So that worked appropriately. Okay. The next one said, create a column of logic called ACE logic. So ACE referring to like polymerase and kinase, and mostly just being used as an excuse to get you thinking about the kind of queries that I do reasonably do. Like, this is usually going to be in a much larger data set. It's usually going to be for probably searching for something a little bit more specific. So like I may be searching for transferases. So we can see here, there's a lot of transferases. If you don't know, a lot of transferring going on in biology, turns out. And so I might just search for transferases and just see what pops up. Or I might search for kinase and see what pops up. But here I wanted to demonstrate some particular challenges when you are wanting to do something that may be attached to a prefix or even a suffix, and it's a little bit more tricky. So here I used ACE, a suffix commonly applied to lots of different types of protein. Since these are descriptions of those genes, they often mention certain proteins. In this case, actually, which is kind of interesting, I will not manage to pull up prenyl transferases because this is plural. And this just kind of gets at why grep and regular expressions in general are often best suited for exploration, not really for the final analysis. Because even this approach that I've taken, so an ACE followed by a space, very fun to say, which matches here, does not match here because that's a plural, and so it ends up not being a match. I also included for the potential that ACE could be at the end of the string, which did actually occur if I remember. Yeah, so like, well, that one's methyltransferases. So I'm missing that one too, but here you go. I would get this transferase with ACE at the end of the string. So I could also have searched for ACEs, space, and ACEs at the end of the string. Oops, I put a space in front of it. That would have caused a problem. If I'd put a space in front of it, that would mean it would have to end in literally space, the word ACEs. Just pretend that that's how that would end up being pronounced. And then the end of the string. So that's not what I want, so this is probably closer. In fact, this is probably the best version of that one I've written so far. Nobody called me on that one yesterday. Yeah. But anyways, so there you go. So this is looking for things that have words ending in ace. There's a lot of ways you can do this to, I guess, perfectly match what I set up here, though. That is, strictly speaking, what I asked you to do. Although this is a better answer for the concept behind the question. Anyways, let me erase that. Okay. So you run that, and then we check our answer. 47. True, true, false, and that one should be false because like that shouldn't contain ace. This had ace in here. And so next year when we use this, I'll have to add some more cases for when they are plural. All right, next up. How many rows contain genes from Panicum Hallei chromosome 2? So here I've used grep. So I've looked at this. I've identified, probably looking through the reading, that this is actually the indicator of the chromosome. I remember I left the answer right there for you, but that's where you would have come up with that. I didn't actually need any of this because if I just look through the data, it turns out that this is sufficiently unique to pull out just the second chromosome genes. But I did just want to demonstrate that, in real life, I often am a little bit more specific because I would often rather miss things as part of like a summative value than to accidentally claim values I didn't have. But it would really depend upon the analysis. Like if this is, just to explore the data, then it'd be important for me to be able to look through everything. And so maybe I actually would leave it at just 2G and then explore what came up. In case maybe there's some other species or something mixed in. If I was for an analysis counting how many results I had in a particular analysis, I might use this very specific one, just to make sure that I don't accidentally overstate the results of my my analysis, because I wouldn't want to be unethical in my research, even on accident. Technically true, kind of a weird example, but that's the idea. Okay, next up, visualize the values inside of the favorite color column. Literally, you can just call table. Pretty easy way to do it. We can see that pretty good spelling this year. I usually get at least a few typos, but really no typos. I had to add a typo that I combined with my favorite color here for rainbow. And so I put some spaces in there just to be kind of messing with you. And then also rainbow, obviously not a single color. So that's not going to work in an analysis trying to figure out like what are like the different frequencies of different favorite color categories. That's not a color, that's a spectrum. So that's going to cause a problem. So that will be part of the cleaning process. I can also see there's some other ones. I can see like turquoise, I can see Tiffany blue, I can see cobalt blue. These are values that may be debatable in what they mean and whether or not they should really just be lumped in with the other blue. We can talk about white and black, if they would be considered here as separate colors. I don't have any grays. I usually get like dark gray as a suggestion, but nothing too crazy this year. But these types of questions are what you legitimately have to ask yourself when you're doing these form of analyses. So this is just harkening back to that slide. We learn all these tools, but the next step, the step of applying it to an actual data set, is bespoke. It's like customized for whatever your data looks like. It's also just challenging because it's different every time. And there are strategies we can use to kind of minimize the replication of work. We'll talk about a few of them, like lowercasing. Lowercase is going to be a really, really common one for string-based analyses, especially anything string-based coming from humans. We just like capitalizing things. I don't know why. There's some other concepts just to be aware of. There is often a issue where there may not be a correct answer. So like metallic blue, they said literally blue, but they also added the word metallic, which invokes a property that's not color, but is perceived as color, right? So it's like specularity. actually multiple colors. It's movements of electrons. It's complicated. I don't know how it works, but metallic blue, not really representing a single color. It's representing other properties of that reflected light. Rainbow, similar situation. Empty quotation marks, different situation. That should clearly be missing or no response given or something like that. But there's a lot of different values that we have to take care of that there may not be a good answer. And so sometimes the answer is simply make it missing, that it was not a data you could interpret, or to code it in some way that informs downstream analyses what happened. So like picked more than one value, or could not commit to an answer, whatever you want to put. But it should be it should allow you to interpret the response correctly, even if there is no good interpretation. It at least can be a correct interpretation, if that makes sense. It's like rainbow, I can't recognize, but I can make it missing, and that is at least a correct, as opposed to making it blue. Obviously, rainbow is not blue. Anyways, account for capitalization, pretty much always. Account for white space, I use trim WS. That's going to help me get rid of these little spaces at the end of rainbow. Just a handy little function for getting rid of white space. So that's like spaces on either side. Make spelling errors that don't change meaning. Oh, no, wait. Make spelling errors not change meaning. I wrote it right. I said it wrong. Turns out no spelling errors this year. That's uncommon. Next up was decide how certain colors should be represented. So that's kind of harkening back to here, but there are often cases like sage green. Well, should that be green? Should it be dark green? Should it be forest green? Should it be its own thing, sage green. Here, I chose to be very specific, to instead of trying to like lump things together, that if they were different words, I split them apart based on this color dictionary. And so like using a reference tool can be a great way to help standardize an experiment. This was getting a little bit like narrowly focused on just color information, so I'm going to skip past this, but it is kind of a cool conversation. Color is fairly challenging, it turns out. I'm coding things as hex values, so that's hexadecimal. That's the value you're seeing here. It's not a base 10, so we work on a base 10 counting system. This is a base 6, I assume, hex. This is the code for the color. Decide what to group or split similar values. So that's what I was saying, like a sage green unique should be coded as green, should be dark green, forest green, what it should call it. Anyways, here we start to explore the data. We can do to lower to make it lowercase, easy enough. We can use trim white space, which is going to get rid of that rainbow's extra couple of spaces. How would you notice? Research, that's it. You could have also done a G-sub. But if you did that, which is fine, you would still need to account for it down here. And that is, something we have to keep in mind. So this would work. We would just later on have to be adjusting our code. Now at this point, you would need to have continued down and found that I had some code to tell us how R interprets different colors. So here's our color check. We can see that the color check is returning that R doesn't know how to deal with rainbow, it doesn't know how to deal with Tiffany blue, doesn't know how to deal with cobalt blue. What I've done here is actually kind of a cool piece of code that is doing 3 comparisons. It's determining if the input values are in colors. It's then determining, are the input values missing? And then finally, it's checking, well, are the input values actually a hexadecimal code? Because R doesn't know how to deal with hexadecimal code, even if it's not part of this large vector of colors. And so I do that analysis, and then I return the results. More on this later. But when I do that, I can see those 3 values were returned, telling me they did come out of this extraction, meaning they failed at least one of these analyses. All right, so then that tells me I need to deal with rainbow. Well, I can't fix rainbow, so I'm going to make it missing. Tiffany blue, I just looked up what Tiffany blue was on that website, and this is what they picked, whether you agree with me or not. This is what they picked. It's on me. Cobalt blue, again, not what I really would have called cobalt. You know what? Nah, that's about what I called cobalt blue. Anyways, moving on. I'm then extracting those values from the data frame and assigning them new ones. And go watch my poll video if that doesn't make sense to you. But what I'm doing here, so I've done like a G-sub. That's cool. Or I could have used trim WS and it's been slightly modified. But then for the actual like replacement, so this being a fairly safe G-sub, no wildcards. Wildcards are dangerous. Here, I am using this whole string comparison tool, so equals equals, and it's only going to affect if the entire value is the word rainbow. So that's very safe. It's not gonna have any extra things. It's not like if someone put rainbow inspired orange. That made no sense. Let's go with the Tiffany blue. So if somebody said Tiffany with 2 N's blue, this would not work, and I would still have to go and fix it, but that's very safe. Like, what if there was a Tiffany's blue, and Tiffany's blue was a different color than Tiffany blue? I don't know if that's true, but it seems like a thing that might happen. I don't know. If that was true, then I wouldn't want to replace Tiffany's blue with this color. I'd want to use some other color. And so in that situation, I would have a second line of code doing that fixing. And you know, some students were asking, can I automate this? And in the end, like you still at some point need a dictionary, right? So you could maybe connect to another website through a tool called an API. You could just create a dictionary yourself, or you could just kind of have done this for several years and just have a list of different things that you tend to apply, which is kind of the approach I've taken. But that's the idea. This is a very safe way, and oftentimes if I do have very complicated string data, there will just be a code file I have that contains how to deal with specific values or specific errors that are super common. I will often use that in conjunction with a G-sub, but with just the caveat that G-sub can be quite dangerous because it is so powerful, because it relies on this partial string magic. Once I've done that though, I now get color check to not be returning anything. That's because all of those values are now either in the colors. Let's make X, just to test this out. They're either in the colors, so all of these trues, or they're hexadecimal values. So all of them kind of fall within one of those two categories, or it's a missing value, which, yeah, just that first one was missing. All right. In the end, if I then find my values, I can see them. If I add use na to if any, I can see that there is still one missing value. Otherwise, the rest of these have been properly cleaned and can now be interpreted correctly through R. I could even put them into like a pie chart, things like that. I could color my pie chart. Maybe I'll do that as the warm-up on Tuesday. Anyways, beyond that, I think I asked you to said which rows contain a gene with the most frequently observed gene family, or protein family. So you would have needed to know that PFAM is your protein family. So these are just ways of classifying different genes, usually based upon ancestry, and ancestry combined with function. It gets a little bit complicated. But there's often multiple ways to define these, or they have multiple domains in them that have different PFAM values, or just, you know, it's kind of creating nuance and ways to classify things. But we can see that each value is actually either a small, so like 68 is a small single protein family, but then 69 has several different values. And so if I take that and I use that string split from earlier, I can split it into a list class object where each container has multiple values. The problem is if I want to calculate which one's the most common, if I take that same thing and I just put it into a table, well, let's see what happens. A problem. It says that all arguments must have the same length, so it's clearly interpreting this list object as multiple arguments. It's not an uncommon issue when sending lists to different functions. So that's a problem. So the answer turns out to be we just need to turn it to no longer be a list. So if it's no longer a list, it's just a big old vector. It's quite a bit longer than that original data frame because some of those values were multiple different protein families. Now at this point, if I call table, I can see that some of these have occurred multiple times. It's a little bit hard to find the one that occurred the most because this is a lot of data, and so I can sort it. Now that I've sorted it, the bottom one is the one that occurred the most. I could have sorted it the other direction. It's not what I chose to do before, but I could go the other direction, and I could call at that point head. Make that head 1, and there it is. And if I really wanted to pull out its name instead of just its value, and please note that that is literally just a value right now, so I can multiply it by 2. Now it's 20. It's not 20, I just multiplied it by 2. But if I wanted just that protein name, I could then just call the names function, and then it's going to pull that out of the attribute of that output vector. How would you know this? Probably by just doing some research or having explored or having seen names enough to have known to guess that that's how you could pull that value out. Or maybe even gone online and searched how to get the name of a value out of a table output in R. A lot of different ways, but that's it. It's the names function. Anyways, once you have that, then if I take that maximum pfam value, which is just that name, I can use that as my pattern in a grep, returning the positions that have that protein family. And let's see if they are at all similar to each other. If they're obviously, clearly they're somewhat similar to it. Let's see if they're obviously similar to each other. And actually look at this ahead of time. That's too much to look at. So instead of extracting by row, let's extract by that definition line. It's a little bit easier to see. There we go, much easier to read. Ah, so that must be a helicase, because all of these are mentioning helicase. Helicase is going to unwind the protein structure, and so that's like remodeling. So how we could probably refer to that. So this is probably also a helicase, or at least involved in like chromosome rearranging. So chromatin remodeling, chromatin being a form of your chromosomes. DNA binding, helicases. Yep, right there. So cool. So we did biology. Whew. Anyways, all right. That took way longer than I guessed. I hope you learned some things. I'm gonna go eat some food. I haven't eaten in forever. Well, a long day. Hey, good luck. Some really powerful tools. Hopefully you're starting to feel very empowered by this, but it does take a lot of practice. There's a lot of different tools, and it's more at this point, now that you have the tool, it's figuring out how to use them. And that's where experience comes in. I can't hand you a garage filled with tools and really expect you to, make me a table. And that's where you're at right now. Like, that's why I'm saying, make me a leg, make me a tabletop, put those together, and then eventually make me a table. But we're getting there. Anyways, y'all have a great weekend. Thank you so much. Probably not watching this today. It's like 6:30 at night. Anyways, bye.